Very good afternoon to one and all. It gives me immense pleasure to welcome everyone to today's introduction program of Early Career Grant of Royal Society of Tropical Medicine and Hygiene. My special welcome goes to the delegates, Dr. George Varghese, Dr. Vinod Skaria, Dr. Shishandu Mukherjee, and Ms. Tamar Ghosh. Thank you for being here and for lending your expertise on this topic. Now I request our director, sir, to welcome the delegates and to say a few inspiring words on conducting the relevant research. Sir, please.
Thank you. Few words, she said. So I'll say few words. Inspiration will come from the people here. Inspiration will come from the heads of the department. Inspiration will go to the students. So that is the mathematical flowchart which I am giving. Welcome to India. Welcome to Delhi AIIMS. And we look forward that this kind of activities will go a long way for all of us, especially, for the people who are at the undergraduate and postgraduate level. And the tag of it, it's not just the money, it's the tag of the where you come from. So that is something which will be definitely an inspiration, and then they should be utilized by all our faculty members, the who are here, and the students, so that more and more competition is there. The people are in line to get this prestigious grants, and then do something which is relevant to the geographical area. Again, that's also very important, that we need to look into our areas of interest which the country is going through, so that we come to the what is there in the mission and the vision of the Royal College, Royal Society, and the Indian needs, and the students have the grant, and then do research what they want to do. With this, I welcome you once again to the institute, and then we look forward that there is a healthy interaction between the faculty members and the experts and the doyens, and we continue to do and see to it that you come frequently and then come with some more grants. Thank you very much.
I request our director again to welcome the delegates. Dr. George Varghese. Dr. Vinod Skaria. Dr. Shishandu Mukherjee. Ms. Tamar Ghosh. Thank you, sir. Now, a brief talk on successful research project from idea to execution will be delivered by Dr. George Varghese, Professor and Head of the Department of Infectious Disease, Christian Medical College, Vellore. He is passionate about translating research from the bench to bedside at community level to combat tropical infections. He is the Fellow of the Infectious Diseases Society of America and Fellow of the Royal College of Physicians. He has many awards and publications to his credit. Sir, we welcome you.
Thank you, Kalavani, for your kind words of introduction. Director sir, distinguished speakers, the respected faculty and colleagues. It's a pleasure to stand here and talk to you. And my memory went back to my first research grant as a junior faculty of receiving $6,000 small grant or early career grant, which actually helped to springboard my research career. And I think this is something which, although the money is a very small amount, this has huge implication in one's research career. So when you talk about research, the research is essentially answering scientific questions which are well thought out. Clinical research is answering questions from the patient's bedside or the clinic. Therefore, it can have enormous impact in people's lives. A clinician researcher derives that satisfaction or happiness of solving problems which one after another which comes to him, and he makes a huge impact in the patient's lives. There are several qualities which we have to have when you try to do that, and what the whole process of developing research starts with the research question. What is your research question? Or how do you develop a research question and a hypothesis? And that's where you start. Then go on to develop the objectives, choose appropriate study design, select the appropriate patients or the subjects and the variables, and you develop the study proposal and the protocol. This is what I would discuss in this session briefly. I know most of you are experts in this, but for the sake of early career colleagues, I would just go through this process, giving you an example from my own research in the early days. So in the year 2000, we were challenged with a set of patients with acute febrile illness and multiple organ involvement. Many of them had hepatitis, ARDS pneumonitis to ARDS, aseptic meningitis, so on and so forth. There were clear evidence of multiple system involvement, and the etiological workup for common multi-system infections were all negative, such as falciparum malaria, leptospirosis, dengue, typhoid, all that was negative. So we didn't know what we were dealing with. I was actually doing my post-graduation in medicine in CMC. And the sicker ones who get into ICU were labeled as sepsis syndrome without a definitive etiological diagnosis, and the milder ones who used to recover were labeled as viral illness. These were not too many patients, but I was not very comfortable to leave it at that. And that's where you begin. You ask a question. I mean, it may be just 2 or 3 patients. So I want you to think about what research question would you ask in that kind of a scenario. Just to give you an example. So when you think of a research question, always think of the 3 elements in that question: the predictor or the independent variable, which could be an intervention, it could be an exposure or a risk factor; the outcome variable or the dependent variable, which is the effect of the predictor; and the study population. These are the 3 important elements which should be there in a well-formulated research question. So you would ask, does the predictor cause the outcome among the study population? Or you may ask, is the predictor associated with the outcome among the study population? The question has to be clear, succinct, and focused, which you can clearly answer. Start with a good idea, which is innovative, feasible, and conceptually significant, which means will benefit the scientific community or the public. Be ambitious but focused. Select 1 or 2 single important questions, not too many. An interdisciplinary project, of course, is more creative and will answer different aspects of the same question, which is actually encouraged. You choose your research question carefully. Address an important and timely research question. It is not enough that you find the question interesting. It needs to be creative, experimentally testable, and not speculative. This is usually a logical extension of the current knowledge, and it should generate important new insight. So they say that a good research question should be finer, feasible, interesting, novel, ethical, and relevant. So the question which I asked myself was, is the detection of rickettsia or aurencia by serology, either IgM antibodies or PCR, associated with fever and multi-system involvement in patients who present with acute febrile illness? And that was the research question. This is when we were faced with this problem in the 2000s. There were no definitive diagnostic tests available for rickettsial infections anywhere in India. As you know, those days we had only Wheel-Felix test, which is a protease antigen cross-reacting with this. Therefore, it's neither sensitive nor specific. So our biggest clues were two. One, some of them turned out to have positive Wheel-Felix, and secondly, we found the eschar in some of those patients. You can see the eschar in the groin of that particular patient. Now we know that this is a huge problem, public health threat in India and several countries in Southeast Asia. About estimated 1 million cases resulting in 150,000 deaths annually, more than 1 billion people at risk in Southeast Asia, the geographic region called Sutsugamushi Triangle. 1/3 of them develop severe disease, including multi-organ dysfunction, and the mortality of severe disease in spite of diagnosis and therapy can be up to 24% in severe disease. So we went on to answer several questions. You may start with one and you start addressing subsequent questions, such as what are the risk factors for developing scrub typhus among the people. So the first question, we actually didn't have the diagnostic tests available in India. So what we did is we did a prospective study, collected about 45 patient samples, sent it to a WHO reference laboratory in Marseille in France, Dr. Didier Raoul's lab, and 28 of them came back positive for Aurencia Sutsugamushi, where all the other etiologies were ruled out. This was published in Annals of New York Academy of Sciences, and we called it as an outbreak at that time. We didn't know what we were dealing with initially. We thought it is an outbreak. Eventually, it became very clear that it was something which was re-emerging. And now we know that it is something which was re-emerging, and it is endemic in our region now. So we went on to answer several questions subsequently. What are the risk factors for developing scrub typhus among the people in Vellore district? What are the predictors of mortality among patients with scrub typhus? And of late, we addressed what antibiotic treatment improves the outcome among patients with severe scrub typhus. So a lot of our initial studies were addressing the epidemiological questions, who is developing the disease, what are the risk factors, so on and so forth. Went on to understand more about the disease and the pathogenesis, including the molecular epidemiology. Again, was an ICMR grant with genotyping the organism in Shimla and districts around in the north, Shillong and districts around in northeast, and Vellore and districts around in the south, genotyping the organism. Went on to look at the diagnostics. And you know that we have good diagnostic tests available now. And of late, a very recent paper in NEJM on intravenous doxycycline versus azithromycin versus combination for severe scrub typhus, where combination therapy is proven to be superior compared to either drug alone, just recently published in NEJM. So when you develop a proposal, the application must be hypothesis driven. The research hypotheses are specific statements that predict the relationship between the variables in a study, and the hypothesis should be innovative or will significantly advance the knowledge of the field. It should increase the understanding of the disease or diagnosis, treatment, and prevention. It should be testable by appropriate methods, and you should have an overall goal, and which is goal of clinical research is to improve human health by generating new knowledge about disease prevention, diagnosis, treatment, or management. This would include developing new treatments or interventions, improving diagnostic methods, understanding the disease mechanisms, evaluating health interventions, so on and so forth. The aim is a broad statement that describes the overall intended outcome or purpose of a research study, and the objectives are specific, measurable, achievable, relevant, and time-bound statements that describe the steps or actions that need to be taken to achieve the overall aim. The overall goal will relate to the purpose of the proposal, to the critical need. It ties everything together or is a unifying hypothesis, and you break it down into feasible aims which are measurable. Try to have 3 or 4 specific objectives at the most, usually a primary objective, a secondary, and an exploratory objective, and your statements will say to determine or to compare, to verify, so on and so forth when you frame your objectives. So always have the SMART objectives, which are specific, measurable, achievable, realistic, and time-bound objectives. Then the next step is the study design. You need to choose your study design based on your research question and hypothesis. I don't have to tell you the different type of studies, but essentially based on whether it is an interventional study or an observational study, which is based on the investigator assigns the exposure or not. You have the randomized controlled trials on one side and the observational studies, the cohort, case control, and cross-sectional studies under the observational studies, which you all know. So the research questions and appropriate study designs, give it a little more thought. And when you are trying to look at the frequency and burden of disease, usually you would choose a cross-sectional study or a survey looking at the prevalence, or a cohort study, which of course is time consuming. It will take much more time. And if you're looking at the risk factors, case control design is a good design. You could also do with cohort. When you're evaluating a diagnostic test, usually a cross-sectional study, same with evaluating a screening tool. And when you are looking at the efficacy or the effectiveness of a new treatment, you look at clinical trials or community interventions, and prognosis, you choose a cohort study. So based on your research question, you choose your appropriate study design. Then you choose the patients or your subjects, and you determine the inclusion and exclusion criteria, the sampling design, how to recruit enough people, so on and so forth. And you must carefully plan your variables. The analytical study, the investigator studies the association between the predictor variable and the outcome variable. Now, you would also have confounding variables. In a randomized study, the confounders will be taken care of automatically. So the clinical trials examine the effect of an intervention on the outcome variable using randomization to control the influence of confounding variables. Now, how do you avoid the biases? The allocation concealment it conceals the randomization sequence, which avoids the selection bias, and it is done during the recruitment of the patient. And the blinding makes the participant or the investigator or both unaware of the treatment received, which avoids the observational bias, and it occurs after the patient is enrolled into the trial. Once you have gone through these steps, you plan your concept proposal. For a small grant, it is something like a concept proposal where you write up a two-page proposal with these important subheadings. The background with the rationale, you state your hypothesis and your specific objectives, you choose your study design and state it clearly, methods including the statistical plans and the sample size calculation in appropriate studies, and the budget summary and the timelines. These components, you write it into a 2 to 3 page concept proposal, and that is what we usually use for a small grant or an early career grant like this. Then you go on to build full proposal, which we won't go into details. So essentially, I was just trying to give you an overview of identifying the research question and hypothesis, formulating appropriate objectives, selecting appropriate study design and methodology, and the components of a concept proposal. I would like to leave you with this quote. When Thomas Alva Edison, who invented the bulb, was asked, you failed so many times, what gave you that courage to keep on, moving forward? So he said, I haven't failed 1,000 times. I have learned 1,000 ways which won't work. So the perseverance is something which is extremely important quality for a researcher. You keep, trying to ask questions and trying to find the answer to many of our problems. Thank you very much for your patient listening.
Thank you, sir, for enlightening us with the steps of a research proposal. Next is My Journey from Clinician to Scientist by Dr. Vinod Skaria. Dr. Vinod Skaria is a principal scientist at the Institute of Genome and Interactive Biology. He is involved in creating novel methods and resources for analysis and annotation for genomes and understanding the functional impact of genomic variations. He is best known for sequencing the first Indian gene, also instrumental in the sequencing of the first Sri Lankan genome and the Malaysian genome. Sir, we welcome.
Thank you very much, and it's a pleasure to be back here. Well, for people who are really not clear where I am from the CSI Institute of Genomics and Integrative Biology. We are pretty much around 10 kilometers from where you are. And in a nice Delhi morning, this is how the institute looks like, and I'm sure you would drop by if you are interested. Well, my previous speaker has pretty much taken the sheen out of what I wanted to start. Last 2 decades, I've done a lot of mistakes, probably 1,001 times. And since I don't have an entire day to speak about it, I'll stick myself to only things that worked. Well, in many ways, I've been fortunate because I studied in an era, and I'm sure a lot of seniors in here or older than me would also appreciate, studied in an era where things were very exciting. And the last part of the 20th century was largely remarkable because of this one single invention, which essentially meant that humans can actually peek into the human body without having to disturb the organism or the organ. Well, I'm talking about the era of exploratory laparotomies or exploratory craniotomies, which was given away by this one single invention. And early in my medical school, my radiology professor, who also incidentally ran a CT scanner, I'm sure some alumni in here would remember, got me excited because this was a very interesting equipment where you can actually put in anything and it could pretty much reconstruct a 3-dimensional image of what you wanted to look at. Now, I'm not really going to talk about CT scans, but I'm really going to talk about what really happened when I was in medical school. Well, I was in medical school at the Government Medical College in Calicut, which is one of the oldest medical institutions and probably one of the largest hospitals until very recently. And midway through my medical school, something remarkable happened. And that remarkable thing was that for the first time, humans were able to understand their own blueprint. And that's essentially the human genome. And that really happened in quite succession with the draft human genome in 2000 and the publication of the entire human genome sequence in 2003. And many of my thoughts was really motivated by my professor in pathology. I really wanted to be a pathologist, who said that this is really going to change what medicine is going to be in the next couple of decades. So I really got interested in this new thing called human genome, and therefore I ended up in a PhD program, not really pursuing an MD program, at the CSI Institute of Genomics and Integrative Biology, a new institution which was created in India, and was also incidentally offering a PhD program for MBBS students. And I worked under Professor S.K. Brahmachari, who was the founder director of this institution. And the institution, of course, has transformed over the last 2 decades into multiple different campuses to what we see as a new campus in southern part of Delhi. Well, also to emphasize that I did a lot of things. I changed my PhD problem at least 4 times in a decade. Eventually, in approximately around 9 years, I could graduate. But nevertheless, a lot of mistakes made you learn a lot more things. And I decided to continue in the institute as a faculty, and that was also very probably a remarkable period in the field of genomics, because that was also the point in time when the sequencing costs were dwindling very rapidly. Now, to give an example or to give a historical point, the human genome sequence was sequenced by around 6 countries, multiple different scientists, approximately $3 billion. And India could not be part of the program, while a neighbor China did indeed sequence 1% of the human genome, because we were poor and we could not afford to foot the bill. But when the sequencing started dwindling in cost, there's also the right opportunity, because small laboratories or Indian laboratories who are poor and underfunded in many ways could imagine to sequence a human genome. And that's also the point in time because we were excited into looking at what the genome could bring. So in 2006, when we started our lab, we started looking at how do we sequence a human genome. But of course, it took 3 years for us to actually do that in India. In December 2009, it was announced in the parliament that India became the 7th country with a capacity to sequence a human genome. Now, of course, sequencing one is not enough. You need to sequence a lot to understand what is really around us. So we collaborated quite extensively, sequencing a lot of genomes around us, including the first Sri Lankan, first Malaysian, first Philippines. We were one of the first founding members of the Pan-Asian Population Genomics Initiative, and very recently we have been venturing into the Middle East, trying to sequence and understand the genomes from the Middle East and Southeast Asia. So in summary, it's not about being able to sequence genomes. We can sequence probably thousands or tens of thousands of genomes today, but the real question is, how do we really apply this in real life applications? And in many ways, patience can actually teach you a lot. So the day when the genome was announced, that night, a patient who himself introduced as a bi called up and said, "What you did was remarkable and you can help us." And what we did remind us was that we could have patients with very hopeless conditions. And this is one such hopeless condition, not lethal, but disfiguring, painful. And this is for dermatologists out here, it's epidermolysis bullosa simplex, an autosomal dominant variety of this condition. Over many months, many visits, and multiple marriage functions, we could actually collect this very large family, around 28 individuals in 5 generations, the largest family for this particular disorder reported anywhere else in the world. Of course, we could find a solution to this family. We made the diagnosis, we made the molecular diagnosis, offered a very cheap, affordable test. And today, in the 6th generation, we have no children born with this condition. And that's really where genomics can really impact people like Bhai. Now, of course, when you put together these numbers are huge. There are 3,000 odd genetic disorders, cumulatively affecting around 70 million people in India alone. Just to put in perspective, Delhi has a population of around 20 million, Delhi and NCR, and that's pretty much 3 to 4 times the Delhi population who suffer from a genetic condition. Many of you would not even realize that you might also have a genetic condition because they might go to multiple physician visits, they go to multiple misdiagnoses, and they lose a considerable amount of time before they actually arrive at a diagnosis. And that really points to the real fact that India is a very large country, and that would mean that approximately 1 in 6 of this world is an Indian. And being 1/6 of the world would also mean that we have pretty much every genetic condition, and probably more than what is documented ever in literature. And unlike the rest of the world, which are the other 1/6 of the world is China, which has quite homogeneous population, we have a much diverse population stratified into endogamous groups because of our marriage practices over the last 2,000 to 3,000 years. And even more importantly, we have a close-knit family structure. That means that every individual in this room would be able to trace their ancestors, at least 3 generations. You know where your grandparents are from. And that necessarily means that you can draw a 3-generation pedigree to start with. And that is really the good starting point of genetic, unlike the rest of the world, which has gone through multiple rounds of world wars, multiple rounds of famine and displacement. We have largely been undisplaced for centuries altogether. So in 2015, with my colleague Dr. Sridhar Chivasubbu, we started this program where we went to clinicians and asked this question: where you see an undiagnosed family or an undiagnosed patient, can we go back and look at the genomes of these individuals in a very systematic way through consenting, sample collection, sequencing, and analysis, and with the promise that we will get back to you whenever we find a pathogenic genetic mutation, and you can act upon it either by genetic counseling, or if there's a therapy available, or at least bring a closure to the diagnostic odyssey for the family. And over 7 years or so, this is now built into one of the largest programs in this part of the world, probably the largest program in the world, today working with around 300 clinicians from 70 major centers across the country. So I'll give you one example probably. That's also because this was done in very close association with Professor Shafali Gulati out here in All India Institute of Medical Sciences. And this news appeared in 2015. A family who has 6 children, 6 out of 8 children with an undiagnosed or mysterious illness, progressive neurological in nature, wanted to euthanize 6 out of their 8 kids. Now, they wanted to euthanize because they didn't have a diagnosis, they didn't have a treatment, and they didn't have hope. Of course, you could find the genetic mutation, and in this particular case, we diagnosed as cystic megalencephaly, or in scientific terms, megalencephalic leukoencephalopathy with subcortical cyst type 1. And of course, MLC1 gene mutations have been discovered before, and also the other founder mutation for this was discovered from the All India Institute of Medical Sciences. Now, apart from this one single patient, what we really did very different was to actually go back to the community and ask this question: if this was extremely rare, would the community have this as a carrier? The answer was even more remarkable, that 28% of the family, extended family or close consanguineous community had this particular genetic variant in a heterozygous form. And that would roughly translate to 1 in around 400 individuals born into this community would have this genetic disorder. So what would that mean? It would mean that you could have an opportunity to diagnose patients in this community. We diagnosed another 4 families. We could, of course, publish this extended large report, making it the second founder mutation for this disorder. Both of them have been discovered in India. And we worked closely with the pediatric neurology department, Professor Shafali Gulati, extensively on this particular family. And today we have a diagnostic in the market, and not in a decade's time, in 3 years' time, we have a diagnostic in the market which can now query both this founder mutation, one in the Nalband community and the other in the Bansal Agarwal community. Now that points to a very important question. When we talk about genetic conditions, we also talk about prevalence of genetic conditions. And there's always arguments among clinicians, what is a more prevalent genetic condition? You ask a cardiologist would say cardiac myopathy or cardiac channelopathy. You ask a pediatrician, the pediatrician will say Duchenne muscular dystrophy. Now, what is really prevalent? How do we really know what is really prevalent? And the real problem is that prevalence is all based on hospital estimates, and that's all based on the fact that the clinician really knows how to diagnose a disease. The very fact is the clinicians don't know how to diagnose a disease, and they need to be taught what or how to diagnose a disease, at least for the very large majority of the rare conditions. How do we do that? And the idea to do that is to essentially go back to the population and ask a simple question. If you could sequence genomes from the population and ask what is really prevalent in terms of pathogenic mutations in the population, can we go back to teach clinicians on what is really, really relevant, what is treatable, and can diagnose these patients much early on? And the fact is, a significant large majority of diseases that we think about, well, it remains untreatable. A small minority of treatments and small minority of patients could actually access life-saving therapies early in life if they could be diagnosed very early on. And I'm not doing some hand-waving. I'll show a couple of examples, and this was built at Calicut Medical College, my alma mater. We found a very high prevalence of primary immunodeficiency conditions. It's a large spectrum of disorders, around 400 genes involved, one of the largest class of genetic disorders. And we went back to the clinic and we started teaching clinicians, pediatricians, on how to diagnose patients with primary immunodeficiency. And of course, these children come with sepsis, they die in the clinic, and of course, many of them do not even get to a diagnosis. So when we do systematic survey and systematic screen, because we know the genetic underpinnings, you can actually create a very large cohort, and in 4 years we have created around 250 patients for this particular genetic condition, one of the largest cohorts in this part of the world. And with the government support, we could create a clinic, we could create diagnostic support, and more importantly, this subset is treatable either to IVIG or through hematopoietic stem cell transplants. And with this government support, a large number of patients today could access either or both of these therapies. But many more important fact is that you could prevent the disease from recurring in the families. And these are families who have lost multiple children because of primary immunodeficiency conditions, because they don't have a diagnosis in time and they succumb to the infection. And today we have a very large genetic screening and counseling program running across the communities where the clinicians could, of course, advise them based on evidence. They could screen them and provide an opportunity to the family through prenatal genetic counseling and prenatal testing. And today we are in the verge of starting one of the pilot newborn screening programs for probably the first of its kind for primary immunodeficiency disorders in this part of the world. And this is another such condition. Of course, I'm sure a lot of people would not recognize this particular condition, or if not even heard about this particular condition, because otherwise in global literature, there's an extremely rare genetic condition. It's called familial adult onset myoclonic epilepsy. The gene was discovered only late in 2019. And the first case, the first set of cases in the series in India was discovered by Dr. Radha Mahadevan from Tirunelveli Medical College. Her lifetime of activity over 2 decades, following up every single patient with this particular genetic manifestation. In 2019, we could identify what is really the cause for this genetic defect. And today we have around 430 patients from just one single community in India. Now, to give a perspective, the global literature on this reports just under 30 cases for this genetic disorder, and just in one single community, we have 400 odd genetically characterized patients with this particular genetic disorder. So in other words, we can actually turn around the diagnosis of genetic conditions provided we know what we are going to look at. And this is what we have been doing through multiple different programs, trying to look at population scale genomes to understand what is prevalent, through Guardian working with clinicians to find diagnosis for undiagnosed patients and conditions. Clinician training has been central to all what we do, and through a very specialized program called GOMED, which is run by my colleague Dr. Mohammad Bhaduk, we actually provide very high-end tests free of cost for many of these treatable genetic conditions which are prevalent in the population. And since they're extremely prevalent in the population, still remain undiagnosed, we could work with the industry to create commercial genetic tests which are now widely available across in many of these companies' catalogs. And of course, the holy grail is to be able to discover therapies for rare genetic diseases, and that's what we have been doing all this while. Now, of course, when I talked about human genomes, every organism on Earth today has genome. There's DNA or RNA, and so does pathogens. And that's also something that has been changing very rapidly over the last half a decade or so, and more so with the COVID pandemic. Now, of course, there are microbiologists out here, and what I'd like to state is that microbiology is also changing very rapidly. And the traditional microbiology is about going to an epidemic and finding out what really caused the outbreak. And typically, this takes weeks, months, if not years. If you really go back to the history of Ebola or Marburg virus, this had taken decades before they could be diagnosed or the molecular mechanism of the disease could be elucidated. And that is largely because you had a very laborious microbiological technique, which includes isolation of the pathogen and of course, observation of the pathogen under the microscope. But if you really go back to the history of 3 years ago with the COVID pandemic, you would realize that if you really go back to the paper which described the COVID virus or SARS-CoV-2 virus, the Wuhan Who Won, you would realize that none of these traditional microbiology techniques were ever used. And what really happened is the patient was admitted, the patient's bronchoalveolar lavage was taken, the sample was sequenced, and the sequence provided the information that this indeed is a new coronavirus. And the sequence was available in weeks' time in public domain, and that really formed everything that we today know about SARS-CoV-2 in terms of diagnostics, in terms of prognostics, in terms of the evolution of the pathogen. So in other words, genomics today is probably a better microscope in looking at emerging pathogens. And in the COVID, of course, we have been very fortunate to have contributed to the scalability of understanding this pathogen. And this started in 2020 with my colleagues' lab in IGIB, where we trialed for the first time in the world this high throughput methodology to sequence SARS-CoV-2. And of course, people have been sequencing SARS-CoV-2 in ones and doubles or dozens. But the idea was to be able to sequence SARS-CoV-2, if not in dozens, in hundreds, if not thousands. And that's what we could achieve in a short period of 3 months. We could actually scale up the SARS-CoV-2 sequencing platform, which now today forms the basis of all the epidemiology that we do for SARS-CoV-2. Every new variant that has been discovered is largely through this particular pipeline we sort of put together. And of course, Kerala was one of the first states to actually put together this surveillance methodology, covering all the districts and involving all the public health units in the districts, 14 medical colleges and public health laboratories. And over the period of a year where we did this, we could actually provide very early warnings about emerging variants of SARS-CoV-2, including the ones which could influence policy, like, for example, double masking was a policy influenced by the sequencing and identification of a new variant, which was spreading very fast. And this could also provide very useful insights into what is really happening and also be able to prepare to see what is or where is SARS-CoV-2 going in the future. And this is sort of a chart which could provide this information because the virus, like any other pathogen, evolves in a very systematic step-by-step manner. And by looking at 2 axes of immune escape and transmissibility, we could pretty much plot where the virus is moving and be able to be prepared and able to be looking for the emerging new variants of SARS-CoV-2. I think I'll stop there and take questions, but before that, all of this is also backed with a lot of genomic education, not just genomic education for clinicians, this is just a subset for clinicians, but also lay public through articles, through online programs, and a lot of digital activity that happens around it. Thank you very much.
Thank you, sir, for educating and inspiring us with your journey. Now, the next talk of the day is Early Career Grant Program of Royal Society of Tropical Medicine and Hygiene by Ms. Tamar Ghosh. She is Chief Executive at Royal Society of Tropical Medicine and Hygiene. She has an MBA from Imperial College London and Masters in Developmental Studies from University of Bristol. Ma'am, please.
Thank you. While the slides load, I want to say a big hello and a thank you to Dr. Sir for extending this invitation to me. And a thank you to all of you for giving up your time and joining us here today. It's such an honor to be here. You've probably noticed from my name, I'm half Bengali, very proudly, half Indian. And in my role as Chief Exec of the RSTMH, I'm on a mission to make sure that we do solve some of these big global issues. And what better place than to be in India? And what better place than to be at AIIMS Delhi and look to all of the expertise and skills that are here to help me on this very big challenge. So I'll start. I wanted to tell you a little bit about the Royal Society first, and then I'll tell you about our Early Careers Grant Programme. Do you want me to find them?
Which one?
This one.
Yes.
I can open it here.
Yeah. no?
Yeah.
Just
Lovely.
Make it full screen.
Thank you. All right. So, a bit about the history of the Royal Society before I tell you about our grants programme. So, we were started a long time ago in 1907, and there's not enough time to talk about the ins and outs and pros and cons of that period of time and why the Society was started. Needless to say, in the decades that have passed, we've been trying to really bring it to life as a global society of people that work in tropical medicine or global health, however that definition is used. And we have members from more than 90 countries around the world. We have a very big network of people that help us deliver our goals as a charity, and they are involved in our journals, in our meetings, in our grants programs, and all sorts of other things. But at the heart of the society, we're a very small team of 8. We're based in London, but as you can see, I try to be meeting as many amazing individuals as I can, partners in our field of work, those that I know can help make the difference and help us deliver our goals. So our main vision, our big goal, shared with, I'm sure, many of you, and my personal motivation in life is about improving equity access to health around the world and eliminating some of the diseases that affect so many communities, and hopefully saving lives in that process. Now, as a society, we fund research, we showcase research, we publish through our journals, and we bring together partners to have discussions and try and bring about change. And the way that we bring together our day-to-day activities and these big visions are threefold. One, it's about our members. All of those members around the world influence us, tell us how we should be working, what we should be focusing on in solving these big problems. And it's their expertise that, secondly, we bring together and we utilize to make a policy change, to fund work, to commission new research, or to showcase a certain disease or a challenge which is causing a blockage to achieving equity in health. And then as an organisation who's been in the sector for so long, we have many partners. I would like to count AIIMS New Delhi as one of our partners, and we look to them to advise us, to tell us what we should be doing better to achieve these goals. And this is one of the reasons why I'm here. I would really love to strengthen those links and to hear from all of you on what you feel the society should be doing more of. So this is a snapshot of the membership around the world. As you can see, they're based in many countries. It's too small, I know, for you to see. There are 102 members of the society in India, which is wonderful, but from my perspective, that's not enough. We need to have more expertise, more knowledge. The more members we have, the more we can be influenced in our work. The scope of our work, this is really difficult to define, but it's around infectious disease and non-communicable disease. It's the big diseases, it's the small diseases that affect fewer people. And it's about all of those contextual points which make it difficult to solve health challenges. It's about climate change, it's about the environment, food security, conflict, and other things. What we do on a day-to-day basis, we have a newsletter running every couple of weeks, which has all of our news, and I'm sure you may have seen that. If not, you can sign up to that pretty easily. We have lots of social media channels running. We publish 2 journals. We run events around the world this year in India, in the UK, Nigeria, Japan, just to name a few. We have these global grants, early career grants. So we give them to early career researchers. The deadline this year, I'm going to mention this date a few times, is the 28th of April, which I recognise is quite soon. But I'm sure that if there are great ideas around campus, you may be able to help, or you may yourself have one to bring them to light so we can see if we can fund them. We also run a website called Grow, and this brings together different opportunities for people who want to get more involved in global health. And we take part in lots of policy and advocacy work with our partners across the world. I won't dwell too much on our journals, but we have 2 scientific journals. The impact factors, one of them is just above 3 and one of them is just below 3. One is more clinical, one is more policy driven. One is hybrid journals, so free to publish in, and one is fully open access. So do please get in touch with me if you'd like to hear more about our journals. I would love to see more research coming from AIIMS Delhi. I completely recognise the fact that we're not the highest level of journal in the world, and researchers here will be publishing at the very high levels. But if you have papers that are relevant, we would love to see them. Okay, so I wanted just to provide a bit of an insight into the support that we provide for our members, just in case this is something that is of interest. We provide travel scholarships for our meetings across the world, particularly for those who haven't attended international meetings before and have an interest in that. And then we have different medals and awards that celebrate success from emerging leaders all the way up to the most senior in their careers. And please consider having a look at these and nominating yourself or someone else that you know. There are not many ways to celebrate the success of all of us. I know in day-to-day life we get on doing what we're doing. If there are people that you feel need recognition, please do put them forward. The Emerging Leader Award, it comes with 500,000 rupee prize. We also improve knowledge, so we have an exclusive members area where we post content. So for example, with our grants program, we delivered a webinar to everyone. We delivered more detailed webinars to our members to try and help them to understand some of the application process. For most of our early career grant applicants, they have not done a grant application form before. They have not written their own budgets before. They haven't decided their own outcomes before in a research project. So we've given them some more guidance. We provide mentorship and training and career support. Obviously, I'm sure everyone at AIIMS Delhi is very well supported, but if there are ways that we can help, we try to add to that. And members get access to the transactions journal. There are many ways to showcase your work. So many of our members ask if they can publish a blog, which goes out in our newsletter to 10, 15,000 people about an area of their work that they're really excited about. Perhaps it's not a scientific research piece, so it wouldn't be published in a journal, but it might be really interesting as a piece of learning. I would love to publish more blogs or do more social media posts around the work and the learnings coming out of AIIMS Delhi. We also have events for different times and people can submit abstracts to them. Our members have a chance to talk about their work, whether it's completed research or whether it's still work in progress. And we have a members directory, so our members find each other across the world working in similar ways and in similar disease areas or health challenges and strike up new alliances and collaborations. Okay, I just wanted to say a little bit about RSTMH in India. I started in my role 6-7 years ago, and it's been my mission to try and make sure that we have more influence coming from India into the society and delivering our work. This is where the expertise is. So I've mentioned we have members across India. 2 members of our Board of Trustees are Dr. Varghese, who you met just before, and Professor Samantare, based in Bhubaneswar. Our country ambassador for India is Dr. Shishendu Mukherjee, who you'll meet shortly. We have country ambassadors in 30 countries, and again, their goal is to influence our work, to try and give us advice on what we should be focusing our time and our efforts on. And we're currently working on a number of MOUs with institutions across India to try and strengthen the partnerships. And of course, for us, that's about the knowledge that we can bring and try and deliver change from. I don't know if Suradeep is in the room. We have student ambassadors across the world at many institutions. In India, we have 1 student ambassador, and that's Suradeep Chowdhury. He's based here. I don't know if he's been able to attend, but we're really delighted that we have him as our student ambassador. And we have global assessors. These are the individuals that look at the grant applications. Now, I've listed here, I know this is too small for you to see, but I can share the slides. I've listed all of the grant awardees from the last 4 years from India. And what you'll see, if you can read this list, is how broad their areas of work are and their projects. This is great. It's a great reflection of all of the work that we should be trying to invest some of our grants in. So these are just a few. There are 21 in total. In 2018, Dr. Devaki Nambiar from the George Institute won the Emerging Leader Award. So that was the first person from India in the last few decades that had won that award, and I was really delighted that she did that. And we've had meetings across India in the last few years and hoping to do more next year. And we have a partnership with DNDI where we bring together early career researchers and early career media to try and strengthen their networks together, to try and encourage researchers to speak more to the media around their research, and to be more in touch with local media for their findings. I mentioned the deadline's the 28th of April, so this is the second. It comes up a couple more times. So these are awards, as I mentioned, they're £5,000 in GB sterling, so equivalent in rupees. They are for a primary research project, which should last up to 1 year. It can be any topic in tropical medicine or global health. And I won't maybe follow this slide order. It can be in any type of project. So whether it's lab-based, translation, implementation, policy, behavioral change, patient pathways, it doesn't matter to us. We just want to provide these grants to those that have not had their own funding before at this level. They have not experienced writing their first research question themselves, devising their first budget, and delivering that from start to finish. This is about finding those incredible new ideas that at this level, we can look at a really wide variety of different issues and see what comes. And we hope that by doing this, we inspire many researchers to just start on the journey and see where they go. So our program is, it can be a scientific research or fieldwork-based research, as I mentioned. Sometimes it's part of a larger project. So it could be that there's an individual part of a research team with a more senior PI doing a research program, and the applicant would apply to us for a grant for a 1-year project which is attached to that bigger research project, a chance for them to do their own study at the same time as this bigger project. They can be based anywhere in the world, and it doesn't matter what discipline or sector they're in. So we have many applicants from institutions like AIIMS Delhi. We have them from NGOs as well. We have them from government and from many different sectors. In 2022, last year, we awarded 205 grants, and they were for people in 36 countries. But we only awarded 9 in India. So personally, I'm on a mission to make that number a lot higher this year. So please do help me if this is something that you will be interested in, or if you know people within your areas, your departments, undergrads, postgrads, please do think of the grants program and please tell them about it. They don't have to be a member to apply for a grant. There are no barriers in that sense. Please do give them the chance to apply if you think it would benefit them. So I mentioned some of the eligibility. I know that some applicants don't get their grants, aren't successful the first time round, and they can always try again. If they've had a travel grant or any other kind of grant, it's also fine to apply to us. And we have lots of PhD or MSc students that apply for our grants every year. So as long as the grant isn't containing containing tuition fees, then they're able to apply, and it's one application per year per person. This is just showing the growth in grant applications since we really started to talk about this more broadly, and more importantly, before we started recruiting the country ambassadors. So their role is to make sure that people are aware of the grants and other activities. And you can see the growth in the applications and the growth in the number of awards. So we're running at 1 in 10. 1 in 10 is not so bad an odds for a global grants program. So I just really want to encourage you to consider these. What you need, or what others that you may know may need to get involved, they need the research idea or question. They need to be able to start and finish the project within the timeline of a year. They need a supervisor that can support them throughout that project and will provide them with a reference, and someone else who is senior in their organisation as a second reference. If you go to the RSTMH website, you can find terms and conditions and the FAQs. We've got a recorded webinar, me talking about the programme in more detail. And there's also the application form is there so you can see it, and some case studies of really good project statements. Just the timelines, which I mentioned some before. The next deadline is the 28th of April. After that, there's a very intense period of assessment that happens at RSTMH, and we hope to then make the funding awards, the grants, by September of this year. The project should start by December and finish within a year, with the reports being due after 3 months. And I just wanted to stress that the whole reason for this programme is to try and encourage people to get involved in research for the first time and try something out that they wouldn't otherwise have an opportunity to do. And so we keep the process as simple as we can. We know that it's the first time for most of our applicants, and we try and support them along the way. So when they get a grant, their membership is paid for either by RSTMH or by one of our other partner funders, and we support them in many other ways throughout their project. So thank you so much for listening, and I hope you'll consider telling people about the grants programme.
Thank you, ma'am, for letting us know about Royal Society of Tropical Medicine and Hygiene and also Early Career Grant. The last talk of the day is by Dr. Shishendu Mukherjee on funding opportunities in India and beyond. Sir is a medical microbiologist and currently a mission director of Grand Challenges India, which is a flagship program of the partnership between the Department of Biotechnology, the Bill and Melinda Gates Foundation, and Wellcome Trust. So it's yours.
Thank you. I think George, Vinod, and Tamar have already covered major part of it, so I will be very short. And I'm just one of the persons who is sitting very near to you. So anybody can walk into me, talk to me, and we can help you out in whatever ways we can to support your innovations. Firstly, I thank Director Sir for this invite today. So thank you very much. The esteemed faculty for being here with us today, and all of you and Tamar for flying down from London. So what I'm going to talk about is the funding opportunities we have within our portfolio, which literally helps you to take your innovation from literally bench to the bedside. So I work with the organization as a Grand Challenge India is housed within an organization called BIRAC, which is a Department of Biotechnology public sector undertaking, and what we do is literally support innovation from literally concept to reality. And we have programs literally supporting you from what you think, an idea, out of the box idea, to validation of an idea, to the stronger validation, taking it for a larger trial, and then bringing it onto the market. So we have programs, all of them. As I said, we are a public sector undertaking under the Department of Biotechnology. And we know in the innovation cycle, it's a very challenging cycle, literally from discovery phase, development phase, and the delivery phase. And we have tried to design program in each one of the phase. And I know within this institute, everybody of the innovators and the researchers and the clinicians are working in one or the other spaces in the discovery cycle or the development cycle or the delivery cycle. And we literally work hands-on with you to take your innovation to the next level. BIRAC, as we call ourselves a funding agency, but only 60% of our job is funding. Rest 30% or 40% of our job is actually enabling innovation. That is, we help you to write your grant applications, help you to defend your grant applications, monitor your grant application to the next level, and then once it is complete, how to de-risk your application and take it to the next level. So we try to work in that phase along with you. And as you know, biotech or the life science sector, especially linked to the clinical sector, is very challenging. Long gestation period, highly uncertainty. Infrastructure is minimal and challenging. The business roadmap is also challenging. Regulatory, IP. So we have created an environment and an ecosystem where we work with you. We have all these facilities inbuilt so that the innovators literally concentrate on the science part of it, and we are there to handhold you to help you out to the next level. You know, Professor George talked about how good is the grant writing, and I think, you know, he literally took you through all the processes. And, you know, we are within the funding agency, look for always a good grant application. We are assessed by how many good grant applications come to the table. And winning a good grant application, as told by George and Vinod's story, if you see, and Tamar's idea, is all comes into one phase, is how compelling story you tell. That is the way to get a good grant application. I think I always give an example in my grant writing workshops and grant clinics, is I compare a good grant to a fairy tale. You know, in the younger days, our parents used to tell us fairy tales. It's always used to start with once upon a time. And I used to tell my mother, just to poke her to read the next line, because the interest used to go on increasing. And it always used to end with happily lived ever after. So I think that sort of feeling in a grant application is very, very essential to, you know, to really push your reviewer to read the next line. That is how I think we work in a funding agency, and we look at almost 1,000+ grant application each year. So I think the way the grant application should come out is how compelling that question is, how strong the unmet need is, and of course the English you write plays a major role in, you know, telling your story, because you are not there, the paper is telling your story, so that field should come out, so that story should come out. And it starts with title also. You know, a lot of times we see long titles in the grant application. Never. I always advise my applicants not more than 12 words. So this is what, you know, it looks at small initiatives, but I think very important to see how we fund. Coming back to what we do is we fund entrepreneurship. We have incubation grants, we have mentorships, and we have you know, incubation setups established across the country. We work on 7 thematic areas, but literally these thematic areas are going on. Vaccine, clinical trials are our main fortes, device diagnostics, drugs, bioinformatics, agriculture, nutrition. We have a very plethora of, this is what I just wanted to tell you, that we literally work from ideation to market. You name a level of innovation you are working in, and we have a program to support you to that level, from literally starting from pre-proof of concept. You have some idea, but you need to validate that idea. You need some small amount of money, and what Tamar's program said, that is exactly what it is. You know, you have an idea, but you need to just test it, that idea in a test tube or a flask or a very small level. You need small grants. We have those grants with us. And we are working with Royal Society to support that earlier career grants also to take it to them. So I will not describe each of my funding schemes, but we have we have programs for each and every of the challenges which you are all working on. These are the level of fundings which we do, so I will skip that. Ignition grant is one of our very flagship grants. I think a couple of your colleagues have won it. Ignition grant, it is an out-of-the-box thinking grant, which you have thinking, you want to validate that idea. 50 lakhs, 18 months. You don't need a company. You can apply in an individual capacity through any of the incubators, and you get this grant, 18 months to validate it. A very interesting grant where you can validate your new ideas. Then we have students grants called Sitare. They want to immerse in some problem, want to take out a grant. We have provisions for them as well. BioNest, and I think we are delighted that AIIMS has one of our BioNests also. These are very unique system. We have 75 during the 75 years of independence. We have established now 75 incubators where we have created all in-house facilities. You can walk in there and with the help of mentors, with help of designing the experiment, the infrastructure, you can validate your concept or the thoughts and the ideas. National Biopharma Mission, and I'm privileged to lead that as well, is another unique initiative which has been established by the Department of Biotechnology and the World Bank. We literally fund in broad areas of literally biopharma products, processes, technologies, medical devices, diagnostics, everything from literally concept to reality. This is a very innovative program. We call it I3, Innovate in India. And this literally funds innovators from all the levels, and there are various levels of grants here. We have specific projects. We have created shared infrastructure. We have almost 35 clinical trial sites developed across this country. And they literally helped us during our COVID challenge to integrate with the mission COVID Suraksha, which also I'll speak very briefly about, is literally support that to create those infrastructure, not only in clinical trials, but bioassays, animal challenge facilities also were created. Grand Challenges India is another initiative which is my bread and butter, and my colleague Richa is also here, so we work on that. We have almost 22 programs or initiatives within that. We work on the HPV vaccine which just came out, and Professor Nirja just left. She was the PI for that. We are working on one of the other projects with her as well to start with. So I think we looked at maternal and child health, infectious diseases, agriculture, sanitation, and we have cross-sectional work as well here, working with basic science clinicians together on policies. So we have a wide variety of programs here as well. The Mission COVID Suraksha, which is I think a well-known program which we managed from our BIRAC as well. We, as you all know, the 4 vaccines which came out from this facility, all the nasal vaccine, the DNA, mRNA, and the subunit vaccine came out from it. We created 35 clinical trial sites. The backbone infrastructure, entire thing was created from this. And right now a call we announced on therapeutics also, which is right now going on. Finally, this will be my last slide, and I would like to tell you that any challenge you make, any program you design, this has to be thought through well on a 360-degree approach. It doesn't mean that at the end of 18 months or 12 months or 24 months you require a product in the shelf. But at least thinking that the program becomes de-risked to a certain level. That's what George also said, that you need to think through that program, and through thinking is not done singularly. And we always promote and tell everybody that you need to build teams around yourself. You are a good clinician, you are a good basic scientist, talk together, build a team, and we have process to fund you and your team together. Thank you very much.
Thank you, sir, for taking us through various funding programs. Now we have come to an end of the program. For open session and closing remarks, Dr. Vijay Mathur, Faculty Coordinator, Clinical Research Unit, Research Section.
A very good evening. After this interesting session, I'm sure you will be having many questions to ask. So let's start with some of the questions. Do we have any online questions in the session chat box? If there are none, we can start with the people sitting here.
Vijay, I think this would be a good grant for MD students, for the guides to inspire, to tell the MD students or DMMCH students, PhD students to apply for this grant, because in a way it would support the thesis, and most of the students do not have any grant at all. They would be eligible for that, and they would have a mentor who can take them through the grant. This would be the perfect kind of placement for this grant, I would say.
I think Kamar can answer that.
Yeah.
Yes, that's right. They would be eligible. And as part of the application process, they're asked to name a supervisor who is with them throughout the project term. And they would act as their mentor and guide and help them. And we ask, because many of them haven't produced the application, that kind of application themselves before, that they work hand in hand with their supervisor to think about the budget, to think about the methodology and the potential outcomes. And then also dissemination. We ask them to talk a bit about what they will do with their findings, whether they're expected or unexpected, and what secondary results might come out of that study, which are always really interesting.
So as I understand that for established researchers who already have grants, they can encourage their students to apply for these grants. For people who are for early career researchers like assistant professors and scientists who do not have any grant, they can readily apply for this.
Yes, absolutely.
Besides that, our directors introduced two schemes. One scheme where we have talked about the intramurals which are available. For the undergraduates, he had taken out a scheme in which every undergraduate would get 50,000 rupees off for doing research. Everybody.
I think we'll have to rotate through him, otherwise we will start giving to everybody.
Earlier, what we had was even for undergraduates, we had a UG mentorship scheme where 2 or 3 undergraduates, they get together, they have a mentor. Because often undergraduates would want to do a summer project. They would walk up to a faculty, we want to do a project. They would like to do a project, but there would not be enough, even sufficient money to buy an ELISA kit or something. So now this supports around 2 lakh rupees for the team of undergraduates who do with the mentor. But thereafter, what Dr. Srinivas has done, that he said that why only few people who apply for it. Research should be a mandate which should be encouraged for undergraduate students because they are actually the time when they're untainted. They are not kind of blinded by any fixed ideas. They are the brightest ideas. So each one of them should be encouraged to do research. So mandatory that each one gets 50,000 rupees of seed money as research. So that during those 5 years, they utilize it and produce for something out there.
One more clarification I wanted, that our post-graduation is normally 3 years, but normally they do thesis which completes in 2 years of time. So seeing this cycle, they may end up either completing it before 1 year, or maybe if they apply early in their start of the post-graduation, they may end up completing their project after 1 year and not within this time period. Then what happens? What are your rules and how flexible it is?
Yeah. So we use a year as a guide for the complexity of the fund that we provide. And we do that because we want them to try and finish quickly and to move on to what the next challenge will be. We do have some projects that need to be extended by a little amount, and we would either decide those at the team, my team, or if we need to, we would talk to our committee. We have a grants and awards committee if there are more complex questions, but we'll try and encourage them to get through the project as quickly as they can. And I didn't mention in the presentation, and possibly this is assumed, but there are no outcomes expected with our projects. We don't expect them to necessarily have a piece of work that can be published or presented at an international meeting. For many of our grant awardees, the biggest achievement they have is confidence in their skills and confidence in what they want their career path to take. And the skills that they learn around budgets, around project delivery, around new collaborations, they recognise as a very high and important part of the project. So in that sense, the pressure is off in terms of what they must achieve through doing this project, this grant.
That's very encouraging. Any questions from the audience?
Hello, am I audible? So I have 2 questions. Is it mandatory to be a member of the society to apply for grant? And second question, is it strictly for only tropical disease and infectious disease or any research related to non-communicable disease?
I don't have enough time to go into the definition of tropical medicine, and I don't like the terminology. It's any topic in global health. Yeah.
Thank you.
Yeah.
So for a PhD student, if they are not employed, they are on a contract for shorter duration. He is asking, can they apply independently or they can apply under the guidance?
Yeah, so there's probably some work to do with the team here. Our grants are for individuals, they're personal awards, they're about their career. When our assessors look at the application, they're looking at the individual as much as the project, and both have to be right. It has to be a grant that will impact their career and they hope in a positive way, and also be a project that they feel will deliver something interesting. And so it doesn't matter if they're employed at the time of applying. We would ask questions to make sure they're supported. They need to have a supervisor and someone else a second reference, but they don't have to be employed.
So in case if a researcher is completing the term in this institute and not getting any financial support or stipend from any of the sources, can he try to get the funding out of this as his honorarium?
Doesn't support salary, I believe.
The support to the research.
Honorarium,
Support only for consumables.
She made it very clear, it is open to all those people. You need not be a permanent employee or something like that. It's an individual basis. It is not necessarily to a tropical medicine, I'm sorry. It is for the people who are done on the behavioral studies. You know, see, one of the topic which she said that what is the behavioral nature of the people who are going for the particular disease, you know. And it is nothing to do with your employment or permanent this thing. What they want is just a supervisor to guide it, because having taken the money, if I can't do the research, and it's going to be waste, you know, that's what they're going to confirm.
Yeah. Yes, right, yeah.
Any more questions, please? For the ambassador.
Was the student ambassador or the country? Student ambassador. So student ambassadors are, they apply through a simple form that's linked from our website. And the assessment really is, do we feel that they would benefit from that role? So generally that's about, do they have a network that they'll disseminate information to? Are they happy to advise us? Because we want to hear from them as much as we want to share information with them, to tell us what we need to be doing to inspire the student career stage. And so, yes, it's open. And we normally, in many countries, we try and have a student ambassador at each institution, because they have slightly different networks, work in slightly different ways. And as I mentioned, in India, we have one, and Suradeep is here in AIIMS Delhi.
Thank you.
I think the benefit for the student is actually two. One, the network you are exposed to. I mean, you get a bigger network of people in the society, in the profession, and also it will look good on your CV. So those are the benefits from your side.
You want to be? Will you? You should apply. Yeah. Apply. Apply.
So the deadline that you gave was April 28th. Was that for the concept note, or is that for the entire proposal?
It's entire.
It's for the application.
Full application.
Yes, I should say.
It's a very short application. When you go through it, not a very
The whole application is like a concept note, just 2 or 3 pages. So that's all.
Any more questions?
How many AIIMS people are getting this year?
Actually, Tamar, we would like to ask you that 9 got from India. How many are the applicants?
Ooh, I don't have that data, but I can share it with you.
Yeah.
My hunch would be, I may be biased, but applicants from institutions like AIIMS Delhi would have a very high chance of having an award. I think that the problem is more awareness of the program. So if we can solve that problem, then I think there'd be a good chance for the caliber of students and faculty here. I'd like to see many awards. So we could set a target for this year if you would like.
So I have a question with Dr. Mukherjee. Sir, is there a provision that our undergraduate students, they can visit your place, see how it works, and that will give them some ideas how it
Absolutely. I think the incubators they can visit, we can definitely plan that out for them. They should go and have a feel of it, and then it will help them to immerse into things and design their concept. Of course, I think a mentor is needed to them, and we can organize that. That is
maybe a trip to this place
we can
can be organized during internship or something. It will help them in.
But we do have an incubator in AIIMS. That is, I think AIIMS is the only government
We should take them to our YNS first.
which has an incubator.
So we have almost 75 bionists now, and we want to make it the number 100 in the next 2 years' time. So that's the.
So I think there are no more questions. Any questions in the chat? Any chat? Yes, please. You would like to say something? Yeah.
Sorry, just a last point. You should have on the desk in front of you our new strategy. And if you have that, it has my email address on the last page. Just to say that I'm really open for any questions or comments or anything after today that you think of, or for your students or others that are not here today. I know it's a holiday week. Do please email me with anything. I'm really happy to help, and anything I can do to support you, I will do.
So thank you very much, each and everyone. We finished exactly in time as planned. Thank you to Director AIIMS, Professor Srinivas. Thanks so much, Dr. Vinod, Dr. George, Dr. Mukherjee, and Tamar. Thank you so much. Thank you everyone for joining. We can have a cup of tea outside before going.
A photo?
Richa, यहाँ से सामने से. Please come. Yeah, I think that will. I used. Come.
Join us.
Please come.
Should.
Please come. Call the CRE people. Please come.
Thank you.
अच्छा रिचा कोई ना.